My name is Ali Nehme, and I have a double major PhD in bioinformatics and in molecular biology. So I have expertise in both wet lab experiments and in molecular and in bioinformatics analysis. In this webinar, I will introduce bioinformatics to you. Then I'll speak about its importance and the different fields of bioinformatics. And I will particularly focus in this webinar on the field of transcriptomics and its toolbox, which rely on to analyze different types of bioinformatics data. And I will give some recommendations throughout the webinar, and some points I will stress on certain things that you need to take care of. What is bioinformatics? As its name indicates, bioinformatics is an interdisciplinary field of science that combines biology with computer science. However, bioinformatics is not just these two for sure, because bioinformatics is mostly rely on the algorithms of mathematics and statistics that form the core field on which bioinformatics algorithms and softwares are based. I need to stress at this point, at this level, that you don't need to be an expert in all disciplines to master bioinformatics. There will always be a starting point or a discipline where you start from, and then you go and prove yourself. So, for example, myself, I started as a molecular biologist. I didn't have any expertise in coding or in statistics. So I started as a molecular biologist with wet lab expertise. And then I started to learn statistical algorithms involved in that are used in bioinformatics, more complex, statistics, like the non-parametric tests, the SAM-R, the lemma, the modeling, the statistical modeling, the FDR, machine learning. Even I started to learn machine learning. I started from wet lab to bioinformatics. Now, this is growing very rapidly, and now, for example, the gene expression omnibus database here. So, if you go to the website, so there are now over 3 million samples, 100,000 data series. So each data set contains multiple samples. So there is a large amount of data available. You need some expertise to go get these data and answer a question of interest by using these data that are available freely on the web. Bioinformatics is a complex field that involves many subdisciplines, as I mentioned. It is not just analysis and statistics. It's not just analysis of large data and comparison between biological conditions. Bioinformatics has introduced new subdisciplines into biology, such as network analysis and systems biology. So it's not the classic, analysis. You have two conditions, you do the experimentation, and you compare between the conditions. Now we have what we call systems biology. You need to analyze the whole system. Statistics now is not just about comparison. It's within the system, how things are going. It's inference. Bioinformatics has really improved the field of biology, the understanding of biology. In addition, bioinformatics, it relies on data visualization and image processing, because there is huge amount of data available. And to be able to visualize this data, you need to simplify the view by data visualization. In addition, the complexity of the data associated with bioinformatics and the complexity and the interconnection within biological systems, the biological systems are interconnected. There's no organ that works on its own. They work together, cells work together, everything work together. It's very complex. This complexity itself has helped advances in statistics and data science to be able to develop complex algorithms that can, answer these complex questions of bioinformatics. Now, going back to biology, what is the importance of bioinformatics? The classic concept of biology is what we call the one enzyme, one phenotype hypothesis, or what we call in the Mendelian genetics. It states that a gene produces one enzyme or protein that is responsible for one phenotype. For example, we have the color of the skin, which is a phenotype. So it depends on a pigment, which we call melanin, and this is encoded by one gene. So the hypothesis that we have the gene of melanin, which produces the protein melanin, and this pigment is introduced in the skin, which will produce the color of the skin. However, this view has advanced with the advancement genetics in the genetics field and in the hereditary field, and they introduced the notion of polygeny, or multifactorial traits. What is polygeny? It means that a phenotype is not regulated by a single gene. A phenotype is regulated by multiple genes, and the overall effect of these genes produces this phenotype. And the multifactorial effects is that it's not only the genes that regulate the phenotype, but also the lifestyle and the environment. It also affects the phenotype of an individual. Let's go back to a skin example. So yeah, melanin is one pigment that is involved in the color of the skin, but there are multiple other genes that are involved in the production of this pigment that are involved in the color of the skin, but also the environment where we have the sun, for example, the strength of the sun in the area you are living, this will also affect the color of your skin. With the advancement in the next generation sequencing, a large-scale data analysis, which we call the omics era, and improvement in the modeling and statistical analysis, the whole notion, the whole concept has changed to a systems biology perspective. We know now what we call systems biology, network biology. So we know now that phenotypes are themselves a complex network of biological processes, and phenotypes that are regulated at a multi-level regulatory network, including genes, which we call genomics. You have the DNA, a mutation in DNA can lead to a disease or a change in a phenotype, a change in the color of the skin. Then we have the epigenomics, which is modification to the genomics without change in the sequence of DNA. So these are if you can call them as additives that change the taste of the food, for example. You have the same bowl with the same ingredients, but you add some spices that change, the taste of the food. This is epigenomics. And then you have the transcriptomics. The transcriptomics is the message that is translated from a gene to a protein, from the stationary phase to the functional phase. Most scientists stating that the transcription of a gene to a transcript or to an mRNA is the bottleneck that regulate gene expression. And then we have the proteins, which are the functional units of the biological system. So the genes, they produce proteins. These proteins that are the ones that produce the effects that show the phenotype. But the proteins themselves might not show the final phenotype. They might lead to metabolites, and these metabolites can lead to phenotype. And you can see also in certain instances that proteins even interacts with RNA species and with metabolites, and they all together produce the phenotype. And this is the field of phenomics to see the interaction between the phenotypes between biological systems. And nowadays we have the multi-omic data integration, which scientists are trying to improve this field. They are trying to integrate data at multiple levels in order to be able to understand biological systems better. despite it will be I think impossible to understand the whole biological systems due to its high complexity, but at least you will get a better understanding of what's going on. Well, this is very important, especially in diseases and treatments. In order, for example, let's say trying to treat a disease, but also you need to prevent side effects of it to the treatment. How to avoid side effects? You need to study the whole system, how this treatment is improving my symptoms, but how it's also affecting my other normal biological functions. So I need to find a treatment that best treats my symptoms and at the same time has the minimal effect on my normal biological functions. Here where bioinformatics comes, it has really improved the understanding of biological systems.